Hello scientists, you're welcome back to the channel. It's always beautiful having you here. Today we are going to discuss about conventional PCR. We've been talking about PCR, which is polymerase chain reaction. So we are going to talk about conventional PCR. That's the first type of PCR. This PCR was designed as far back as 1983. 1983 by Karimulis. Karimulis was the scientist that designed this method. And like we've said in our earlier videos, that your PCR is a laboratory technique that helps to make millions of copies of a targeted sequence, a targeted DNA sequence, or a targeted gene sequence. If we want to make many copies for reasons such as studying this sequence very well, we do PCR. Now, the conventional PCR is the method that was designed as far back as 1983, where we use it to make many copies of a gene. And when we are making many copies of a gene, what are the things we do? Number one, we denature the DNA. We know that DNA is a double stranded helical structure, a double stranded helix. So we want to denature it, meaning we want to separate that double strand into single strands. Now we have the area that we're interested in, the gene we're interested in, the sequence we're interested in. We refer to that as the template DNA. That is the area for which we're interested in, that we want to make many copies of what is sequenced in that area. So when we denature the DNA, the next thing that we do is that we anneal the primers. Annealing of primers means that we have what we call primers, which are short DNA sequences. And these short DNA sequences, they are important because they are able to target specific regions. The idea is that a gene has a place where it starts and where it stops along the length of a DNA. So you have a DNA along its length. The gene that you're interested in, it has a place where it starts and where it ends. Now you have primers, which are short sequences of DNA, which when you add them to your solution, they are able to look for that particular target place. That is the place where the gene starts and ends. These primers, they attach themselves to this area, and that's what you call your annealing. And after your annealing, you now have your extension. And what is in your extension? Now you have nucleotides. You have small nucleotides that are now added. These small nucleotides, we call them deoxynucleotide triphosphase, DNTPs. They are now added and they are now used to make a new strand. So the idea is like you have two strands, you unzip them, you take one, and then you use that one as a template to make many strands of that single one, that one that is a template. You make million copies of that one. And you have your primers that are attached to the different ends to signify that, okay, this is where the gene starts, this is where it stops. And then you have your DNTPs, your nucleotide triphosphates that are now added, the nucleotide triphosphates that are now added to that your template strand to make new strands. And also helping to run this process is there are different enzymes, but one of the most important is the Taq polymerase, which is an enzyme that helps to make many copies of this DNA. And this is really important. You also have a buffer solution to make sure that there's no change in pH, and then you have essential cofactors like your magnesium 2 plus, magnesium ions. They act as cofactors, trying to make sure that the reaction goes well. So this is what happens when you do your conventional PCR. And in the conventional PCR, you have, like I said, denaturing, you have annealing, you have extension, and they all occur at different temperatures. So if it's denaturing, it occurs between 94 and 98 degrees. If it's annealing, it occurs between 50 and 56, and then extension occurs at 72 degrees. And then usually when you do the conventional PCR, you do it in cycles from about 24 to 48 cycles. It runs many times. And in this whole process, the idea is that many copies of our gene of interest, of our target DNA, many copies are made. And that's process that you call the amplification process. So this process is important and is the basis for many things, diagnosis, many things that are done in the area of life sciences are done using this method. So it's important to understand the method. It's important to know what you're doing. You're trying to make many copies of a particular sequence of DNA. And in order to make those many copies, you need to know where does the gene start from? Where does it end from? Where does it end at? Where it starts from, where it ends at, you have primers attached to those areas of the gene, and then you have DNTPs being added, to the template strand to make a new strand, and you have Taq polymerase helping to run this process. So it's quite easy to understand, and I hope you have an idea. When next we meet, we'll talk about real-time PCR, and that's how we'll continue talking nested PCR, quantitative PCR, all the different kinds of PCR. Thank you for listening. Remember to like, share our content, and do have a beautiful day.